content editor at GenomeWeb, and I'll be your moderator today. The title of today's session is Considerations for Selecting Saliva Collection Devices for Genetic Testing and Molecular Diagnostics. This e-case study will feature two brief presentations from Austin Udakor, Director of Regulatory Affairs at DNA Genotek, and Dr. Tara Crawford Parks, Senior R&D Manager at DNA Genotek. Special thanks to our sponsor, DNA Genotek, for making this presentation possible. Austin, please go ahead.
Thanks, Colin, for that. If you're listening today, you likely have an interest or use IVDs for molecular diagnostics, such as a genetic test. Today, we'll be touching on this constantly evolving situation with the US FDA's proposed laboratory-developed rule in the coming slides. But first, I wanted to outline a few key differences between IVDs and LDTs for clarity to anyone who may not have foundational knowledge on this topic. IVDs are in vitro diagnostic devices or tests and are regulated by FDA and classified by risk level. With FDA's oversight, non-exempt IVDs must undergo FDA pre-market review where analytical and clinical performance validation data is reviewed. These IVDs must receive FDA clearance or approval prior to being commercially marketed, sold, or used by anyone for or more laboratories for diagnostic testing. On the other hand, lab-developed tests are different from IVDs in that they are regulated by the Centers for Medicare and Medicaid Services, also known as CMS, through the Clinical Lab Improvement Amendments Act called CLIA. This is all in the US, just to be clear. LDTs are currently exempt from FDA oversight. LDTs are generally developed and run in a specific lab and do not require pre-market review prior to commercialization or commencing testing. Instead, there's a requirement that analytical and performance verification of lab processes are completed prior to running the test. Most LDTs, and I'm generalizing here, are developed but not sold commercially outside of the laboratory. At present, the US FDA has applied enforcement discretion over regulation of LDTs. Now, what that means is that FDA does not require LDTs to seek clearance before they put on the market. FDA's enforcement discretion has been a closely watched topic for several years. Currently, FDA has announced that they intend to regulate LDTs and put forward proposed rules. The proposed rules are intended to merge LDTs and IVDs under one device regulation. FDA has proposed a 5-stage transition over 4 years, with prioritization going towards high-risk tests. This high-risk test will now require FDA approval within 3.5 years, but this will not happen before October 1st, 2027. Low and medium-risk tests will follow shortly thereafter and will have to be FDA registered or FDA cleared within 4 years, but not before April 1st, 2028. It is worth noting here that these states are proposed dates, so they are subject to change based on comments that the FDA receives through the comment period. Speaking about the comment period is open until December 1st, 2023, if it is not extended. After FDA reviews, all received content will be reviewed, and a decision will be made regarding the next steps for this rulemaking. This decision could be significant for current lab-developed tests. As I said before, a key difference for LDTs is if these rules come into effect, they would now require free market review, which they do not have to comply with at the moment. In clear terms, that means they will need to obtain FDA 510K or FDA PMA approval before they can put their tests on the market. As every rule goes, there are exceptions. There are certain exemptions for HLA tests, forensic use devices, and others that may come up as the comment period unfolds. If you're a test developer or manufacturer and you're considering your next steps, there are a lot of things for you to consider as you think through how to navigate these potentially new regulations. The goal of this webinar is to help educate on those considerations. overall, you should have included in your strategy some of these points on the slide. By no means, these are not all the things you should consider, but these are just some of the few things we think we wanted to highlight today. Beginning with your regulatory strategy. The regulatory pathway consideration will, of course, be the thing to consider, and it's important to identify which classification your test falls into. You'll also want to think about what the device is used for. This is generally referred to as the indications for use. It's specific to the device or test and identifies the target market. For example, it may be a test for a specific genetic molecular diagnostic or a test for a broader population. Then comes the process for analytical and clinical validation. This isn't something currently required under the CLIA LDT process. However, it is a critical component within FDA's pre-market review process. Essentially, what is the evidence you're going to provide to FDA to demonstrate that the test does what it claims to do and that you have the data to support it? In the example of a genetic test, this would be the validation data supporting an accurate diagnosis for that specific genetic condition or disease. Outside of these high-level considerations, fall other important considerations such as what stage an LDT might be in. For example, if a test is currently being developed, there are considerations to be made on whether the test goes on the market prior to the new rules coming into effect or after. That means that the approach you take now could impact what happens in 3.5 years, depending on the outcome of FDA's guidance. One key area that you should focus on is resources and expertise. Depending on what happens in the next few years with the regulation, ask if you have the resources and expertise within your organization or outside to be able to navigate these new rules. If you feel like you would be struggling, it's a good time to start planning now. On the slide, I'll just show you a couple of examples of LDTs, of FDA-cleared devices that have been used in FDA-cleared tests. For example, we have the Oragene DX collection device, which is FDA-cleared for prescription and direct-to-consumer use. Now, FDA has classified saliva collection devices, such as the ones on the screen, as Class 2 medical devices under the product code OIJ. This is just a code for DNA specimen collection for clinical testing. These saliva collection devices received FDA clearance after FDA review, extensive analytical validation, and clinical performance data. They were used in conjunction with the test you see on the screen to demonstrate end-to-end approval for the devices. Essentially, if the LDT's rule goes to be, this is what you would expect for most tests on the market. There would have been a pre-market review and approval prior to getting on the market. And I'll pass it on to Tara. Tara?
Thanks, Austin. As Austin said, both the Oragene DX and ORAcollect DX devices that we manufacture have been robustly validated through this and reviewed through that FDA process for the 510K. Today, I'll walk through some of the ease of use data that we collected to support these products. Specifically, our studies were conducted to evaluate the use of the products across naive user populations, reflective of the US general population. These studies were designed to evaluate the comprehension of the instructions for use and compliance with returning the collected saliva samples back to the laboratory through the regular mail service as a way to represent use in an at-home self-collection environment. As an example of the type of data we collected during these various usability studies, I will walk you through some of the key findings of representative studies for both the Oragene DX and the ORAcollect DX devices. To evaluate the use of the Oragene DX device in a DTC environment, naive participants were recruited and provided a sample study kit containing Oragene DX. They were instructed first to register the kit, to collect a saliva sample following the instructions for use, to complete a comprehension questionnaire on the instructions for use, and to return the collected sample to a processing laboratory via standard postal services. On the right-hand side of the slide, you'll see our general collection steps for the Oragene DX device that were included in our usability study. The individual is instructed to spit into our tube, which contains a funnel on the top. They then close the funnel, and this releases chemistry that is contained in the cap. They then remove the funnel and place a cap on the tube, following some mixing steps. And then they are instructed to follow shipping instructions back to the processing laboratory from our study, where they place the collected saliva sample back into a biospecimen bag and return it in a mailer box. Once the samples were received at the lab, DNA was extracted and evaluated for our study criteria, such as DNA yield endpoints. And then it was processed, these samples were then processed on a downstream molecular diagnostic assay. The study results demonstrated in our study that greater than 97% of the collected samples met the DNA yield criteria, and 96% of the samples met the performance criteria on the downstream genotyping and DNA sequencing assays. In addition, more than 96% of participants agreed that the Oragene DX instructions were easy to understand and follow to correctly collect their sample, while 99.5% of donors understood how to follow the shipping instructions to send the sample back to a processing laboratory. Similarly to the Oragene DX usability study described in this last slide, the ORAcollect DX study recruited naive participants, and they were provided a study kit containing the ORAcollect DX device. Participants collected a sample following the instructions for use, completed a comprehension questionnaire related to the collection instructions, and followed the provided shipping instructions to mail the sample back via standard postal services. On this slide on the right, you'll see the collection steps for the ORAcollect DX device, where a user is instructed to insert the sponge into their mouth and collect the saliva sample. They then remove the cap from the tube and invert it, and this swab goes into the tube that contains a stabilization chemistry. They then follow shaking steps to mix the sample appropriately. And similarly, as what I described for the Oragene DX study, they then were instructed to put the saliva sample into a biospecimen bag, follow standard shipping instructions, and mail the kit back to a processing laboratory. The results from this study were very similar to the Oragene DX study. More than 95% of the study participants understood how to collect their saliva sample and to return it to the lab for testing. When tested through the molecular diagnostic assay, 99.5% of the DNA samples provided the correct result. In addition, 95% of participants answered the comprehension questions correctly and complied with the provided shipping instructions. Overall, the results obtained from these two studies support the ease of use of both the Oragene DX and the ORAcollect DX devices and highlight the potential to use these devices to support sample collection workflows, particularly in a direct-to-consumer environment. Going back to Austin's earlier point on considerations around timing or time to market, leveraging devices with FDA 510(k) clearance can provide significant benefits when it comes to time, effort, and cost when validating, for example, a molecular diagnostic test for marketing authorization. In addition to time to market, additional important aspects to consider, more so for a commercially marketed test, are a streamlined end-to-end operational workflow and brand recognition. This is of particular interest, for example, for genetic testing companies whose target market might be a consumer who is interested in their genetic predisposition for a particular disease. In this regard, flexible customization services, which include design and branded materials such as custom instructions for use with the company logo and color palette, and custom branded packaging options can enable companies to create a solution that serves the entirety of their workflow. In the customization options for Oragene DX and ORAcollect DX devices, as shown here, for example, you can see the various user touchpoints where branding can be placed. In addition to branding, the customization options extend down to the saliva sample itself with customized barcoding solutions, including 2D barcodes, QR codes, and accessory barcode labels to provide donor privacy while linking individuals to their collection devices to track the sample throughout the entire chain of custody, from shipment to the donor's home or clinic and then back to the laboratory for processing. For companies requiring logistical support to enable their diagnostic tests, single order fulfillment or kitting and shipping services are also available. We hope these insights were valuable for you as you navigate the new landscape of regulatory requirements. We understand the importance of a trusted collaborator who combines technical acumen with deep regulatory experience, servicing test developers. Our consultative approach tailors solutions to the individual's needs of test developers, whether they're looking to switch to a saliva collection device with general clearance by the US FDA, or looking to streamline their transition from an LDT to an IVD. For more information, please contact us at info@dnagenotech.com. Thank you for listening, and I will now pass it back to our moderator, Colin.
Great. Thank you, Tara. Thank you, Austin, for your presentations. And thank you to our sponsor, DNA Genotech. And thanks to all of our viewers for joining us.